Welcome back. So now we're moving on to really talking about what makes life possible. So we laid the foundation. We talked about all matter and what is matter made up of. Matter's made up of atoms and molecules and compounds. Well, all of these things, while they make up everything in the universe, all right, when it comes to living things, all right, not all atoms are part of living things, not all molecules are part of living things, but all cells are going to be part of living things. So what deciphers something from being living and non-living is that they are made up of cells. So if it's made up of cells, we know it is a living thing. So the cell is the smallest unit of life. So while we talked about atoms are the smallest unit, and they're that, and it's made up of subatomic particles. Um, Really, atoms are the smallest, um, really functional thing within matter. When we start talking about what is the smallest unit of a living thing, we're talking about cells. So the cell is the smallest unit of life or living things that can function independently and perform all the necessary functions of life, including reproducing itself. So all living things are made up of cells and they can reproduce themselves. So when we are trying to break this down into something that, as far as trying to organize things, we really start at atoms. And atoms are going to make up molecules. And then molecules can make up larger molecules, and those larger molecules can make up organelles. And organelles are going to make up cells. And this is where we start to consider things to be living. And then cells, of course, can make up tissues. And tissues are going to make up organs. And organs are going to make up organ systems. And organ systems are going to make up an organism. But if something isn't made up of tissues or organs or organ systems, it can still be a living thing. A single cellular organism is just made up of one cell, but it's still considered a living thing. So we talk about in biology the cell theory. Well, what's the cell theory? It's this theory in biology, and it's accepted by all biologists. And it states that all living organisms are made up of one or more cells, which I just said, and all cells arise from other pre-existing cells. So we know that living things are made of cells, and cells come from other cells. So prokaryotic cells are structurally simple but extremely diverse. So the early cells, we're talking about billions of years ago. We're talking about very simple cells. So cells, we always talk about cells, and if you took high school biology, you usually see a poster on the wall of a complex cell with all these different organelles. But once upon a time, and they still exist today, but there used to only be these prokaryotic cells, and they were much simpler. They didn't have all these organelles that made them more complex. So prokaryotic cells, they're very simple, but they're extremely diverse. There's many different types of prokaryotic cells. So every cell on Earth falls into one of the two following categories. They're either going to be a prokaryotic cell or they're going to be a eukaryotic cell. Now, prokaryotic cells are very simple, and the eukaryotic cells are much more complex. And eukaryotic cell has a central control structure, which we call the nucleus, that contains the cell's DNA. So uh, a eukaryotic cell is going to have a nucleus, and the nucleus is going to be the kind of the brain of that cell or the control center of that cell, and within it is going to be the DNA, which is the instructions. And we call things that contain eukaryotic cells eukaryotes. And then you have prokaryotic cells, which are going to make up prokaryotes. And with one way to really tell the difference, not only do you see that eukaryotic cells have these organelles and prokaryotic cells don't, you see that when you look at a prokaryotic cell, it doesn't have a nucleus. It still has genetic material, still has DNA, but its DNA is just within the middle of the cell. It's in the cell, floating around. It is not enclosed in a nucleus. So they share four basic structural features, and you're going to see that they have a plasma membrane, which is going to be this outer portion, and a cytoplasm, which is going to be the liquid on the inside. And then you're going to have DNA floating around. And then you're going to have ribosomes, which are able to copy the DNA. So that's really all it is to it. You don't have all these different organelles like you see in your biology class. But you can also have a cell wall. There can be a capsule as a protective coating. You can have pili, and you can have flagellum. So you can have these kind of small hair projections, and then you can have these flagellum, which is going to be there for locomotion or movement. So eukaryotic cells have compartments with specialized functions, and you see that these compartments are known as organelles. Okay, the typical eukaryotic features are going to be a nucleus that has the DNA in it, cytoplasm, which contains the organelles, and then they're going to be larger than prokaryotes, usually at least 10 times larger than prokaryotes. So they're a lot larger, they have organelles, and they have a nucleus with DNA in it. And opposed to the prokaryotic cells, which have no nucleus. They have DNA, but no nucleus in it. Internal structures mostly organized into compartments, and mostly not organized, into compartments, and they're much smaller than the eukaryotes. So this is an animal cell, which is a type of eukaryotic cell. So you have prokaryotic cells, and then you are going to have eukaryotic cells, and eukaryotic cells can be animal cells, plant cells. So this is an example of an animal cell. And if you notice, animal cell is depicted as this round cell, and it has a nucleus and usually a nucleolus inside it. And within the nucleus, you're going to have the DNA, and you're going to have other organelles as well. You're going to have an endoplasmic reticulum. It can either be rough endoplasmic reticulum, which has ribosomes attached on the outside, or it could be smooth endoplasmic reticulum that does not have the ribosomes on the outside. And then you have a Golgi body or Golgi apparatus, and you have ribosomes, free ribosomes that can float around. You have mitochondria, which are the powerhouse of the cell. They're there to make energy for the cell. You have centrioles, and then you have other accessory organelles, which include peroxisomes, lysosomes. You also have cytoskeleton, and then it's all floating around in the cytoplasm with the cell membrane on the outside. On the other hand, you have also another type of eukaryotic cell is a plant cell. So plant cells are usually drawn looking kind of rectangular. This one is kind of like a hexagon, and you see that it doesn't look very round. It looks like it has these flat portions to it. And a lot of the organelles are very similar, except you'll have things like a cell wall, okay, and you can have chloroplasts. They're there for photosynthesis, which animals don't do. And then you can also have vacuoles, right? Sometimes you'll find those in animals, but they're not as big as this. So you see how massive a vacuole is in a plant cell. So if you ever see, what's the difference between a plant cell and an animal cell? Well, we know that if we can find a chloroplast in there, it's got to be a plant cell. If it has a cell wall, it's not going to be an animal cell. And if it has a very large vacuole, that's going to say, hey, this is a plant cell as well. So we have something that's called the endosymbiosis theory, and we'll talk about this later when we start talking about evolution. But endosymbiosis theory provides currently our best explanation for the presence of chloroplasts in plants and mitochondria in plants and animals. So in eukaryotic cells, you can have chloroplasts and you'll have mitochondria, and these are very important organelles, but these organelles are rather complex, and their complexity points to the idea that they might have been their own type of organism before. They might have been their own type of organism, and they went into the cell and they kind of became part of it. So we often talk about symbiosis. Symbiosis is two organisms living very closely together. So something like the bacteria that lives in your gut that helps you and you help it. Um, Finding Nemo, you have the clownfish and the anemone. They live very closely together. That clownfish needs that anemone needs that clownfish. So that word symbiosis means living very closely together. And endosymbiosis is the idea that it is going inside the cell and it is creating that relationship. So symbiotic relationship. So according to the theory of endosymbiosis, the two different types of prokaryotes worked in close partnerships. One went into the other, and that's how it eventually made these eukaryotic cells. So the theory of endosymbiosis is supported by several observations. One is that chloroplasts and mitochondria are similar in size to prokaryotic cells and divide by fission or splitting. Both chloroplasts and mitochondria have ribosomes of their own. Chloroplasts and mitochondria have small amounts of their own DNA, which is circular shape. And chloroplasts and mitochondrial DNA are more highly related to bacterial DNA than they are to eukaryotic DNA. So when we talk about endosymbiosis versus sometimes you'll hear this term invagination. Well, how are they different? Well, you see in um, endosymbiosis, the ancestral eukaryote engulfs the prokaryote, all right, and then they're going to merge, and over time, the engulfed prokaryote evolves into an organelle, such as the mitochondria, and now they're kind of stuck together because they need each other to live. Where in invagination, you see that the plasma membrane is going to fold onto itself, okay, and then it's going to create these organelles. So one, you see that something is becoming part of the cell, and this, it's really the cell is forming these organelles. So we have these two things called endocytosis and exocytosis used for transportation of particles. In endocytosis, you have absorption of large particles into the cell, where exocytosis is going to be moving large particles out of a cell or exporting it. So in endocytosis, you're going to have engulfing large particles using the plasma membrane. And there's three different types you're going to hear about: phagocytosis, pinocytosis, as well as receptor-mediated endocytosis. And phagocytosis is when you're going to see that a cell is going to really kind of create an opening for it to consume something. All right, so the plasma membrane forms a pocket-like enclosure around a large particle, and the particle is going to be transported into it. Opposed, on the flip side, you have receptor-mediated endocytosis, where it's a little more complex. You have these receptors that are going to pick up on what type of particle that it wants on the inside. So you have an example like LDL, which is cholesterol, and LDL or cholesterol receptor proteins in the plasma membrane of the liver cells recognize and bind to LDL. So they're able to pick up on, hey, there might be all these different types of particles going by the liver, and the liver cells have these kind of markers that are going to pick up on which type of molecule it is, and if it's not cholesterol, it's not gonna let it in, but it's gonna pick up and say, hey, you know what? This cholesterol, I know that this is cholesterol, and I'm going to let it in. So it picks up on that with the receptor. The plasma membrane is then going to form a vesicle or kind of create a pouch for it to go into, and then it's going to engulf that cholesterol, that LDL, and then the LDL is broken down and used to make the molecules that it needs for LDL. So as much as you hear about people saying, "Oh, LDL is bad for you," cholesterol, the LDL is considered the bad type of cholesterol. As much as people say that is the bad type of cholesterol, um, you do need cholesterol in order to maintain a balance and to function. So it can be used elsewhere in the production of steroid hormones, including estrogen and testosterone. So we need those things. So not completely bad, but you just don't want too much of it. So faulty membranes are a primary cause of cardiovascular disease. You see that, in a healthy artery, it's going to stay open, but you create, if you have too much LDL, or when circulating cholesterol levels exceed the capacity for binding, or there's an issue with those receptors, then you're going to have this buildup, and then blood flow can be reduced or blocked. Because it's not removing that lipid and it's forming that plaque there and that buildup stops the flow of blood, or reduces the flow of blood. So you see, you have this healthy artery here, nice and large, plenty of blood can go through it, and then you have this really, narrowed artery that is going to be blocked. And then you have exocytosis, which is going to move molecules out of the cell, not limited to large molecules necessarily. Vesicles first enclose the particles, so it really makes a pocket to contain everything, and then it merges with the plasma membrane, and it pushes it out. So it very similar. It's kind of the reverse action of what endocytosis does. So connections between cells, when we're talking about cells being connected, you talk about tissues, different cells coming together and working together. There's different types of connections between cells that are going to hold them in place and enable them to communicate with each other. So you have three different types. You have either tight junctions, desmosomes, or gap junctions. And in tight junctions, they form this watertight seal between the cells. In desmosomes, they're kind of like Velcro coming together. And then in gap junctions, they act like this secret passageway, which allows certain materials to pass between the cells. The nucleus is the cell's genetic control center, or the kind of the brain of the cell, and it's the largest and most prominent organelle in eukaryotic cells. And the nucleus has two primary functions. It's going to be the genetic control center, and it's the storehouse for the DNA, or the hereditary information. Some structures of the DNA of the nucleus. It acts as you're going to have the nucleolus, and you're also going to have chromosomes, or chromatin in there, which is the genetic information. And you also have a nuclear membrane, and the functions of the nucleus are to be that control center, but also to contain those chromosomes, or that genetic information. Cytoplasm and the cytoskeleton form the cell's internal environment, provide support, and it can generate motion. It's what all the cytoskeleton, or I'm sorry, the cytoplasm is where all the organelles are floating around. Cells can have cilia, flagella. Cilia are short projections, and then flagella are going to be long ones that are there to move the cell, opposed to cilia, which can move fluid along and pass the cell. Mitochondria, energy converters. All right, once we consume glucose, we have to turn glucose into ATP, and that is the responsibility of that is the mitochondria. So the number of mitochondria in cells varies depending on the what cell type it is. They're more densely packed in cells that have high energy requirements. So you see something like an energy, a liver cell needs a lot of energy. It's going to have a lot of mitochondria. And then you have a skeletal muscle has a lot as well. With endosymbiosis, mitochondria may have existed as separate single-celled bacterial-like or prokaryotic organisms billions of years ago. So we're talking about something that was like a prokaryote, and it made its way into a early eukaryotic organism, or cell, and that's when the two became one. So mitochondria actually have their own DNA, and all the mitochondrial DNA is descended from your mother's mitochondria. So they actually, everyone gets their mitochondria from their mother. So no matter who you are, you have the same mitochondrial DNA as your mother does, and her mother has the same as her mother, and her mother had the same as her mother, and you can go all the way through the female's lineages of your family. Whether you're a male or a female, you can go through and see all the females there were before you, because they all had the same mitochondrial DNA. Now, lysosomes are the cell's garbage disposals, and they kind of act like this kind of floating thing that is going to digest and recycle cellular waste products and consume material, break them down, make sure that we get rid of them. In the endoplasmic reticulum, you have cells build proteins and they're going to disarm toxins. In the rough endoplasmic reticulum, you're going to see that proteins will be shipped to other locations, um, and um, the rough endoplasmic reticulum is called rough because it looks kind of bumpy. It has ribosomes, which are protein-making machines, on the outside of it. Smooth endoplasmic reticulum, it's going to synthesize fats, lipids, such as fatty acids, phospholipids, steroids, and it detoxifies molecules such as if you come in contact with this, if the cell comes in contact with alcohol, drugs, or some other kind of waste. And then you have the Golgi apparatus, sometimes called the Golgi body, um, processes products for delivery throughout the body. So kind of a processing center that's going to transport things, which is made possible from the Golgi apparatus and these transport vesicles. So when we talk about the endomembrane system, you have the transport vesicle buds from the smooth or the rough ER. Transport vesicle fuses with the Golgi apparatus, dumping contents inside. Then you're going to have the Golgi apparatus modify the molecules as they move through its chambers, and then the modified molecules bud off from the Golgi apparatus as a transport vesicle, and the vesicle may fuse with the plasma membrane, dumping contents outside of the cell for delivery elsewhere in the organism. So you see where it starts with this smooth or rough endoplasmic reticulum, and it sends it to the Golgi apparatus, and the Golgi apparatus, it kind of prepares it to then send it outside. The cell wall provides additional protection and support for plant cells. So remember, cell wall, large vacuole, chloroplasts, all these things are part of plant cells, not so much animal cells. Plasmodesmata are these tube-like channels that connect plant cells to each other. You have vacuoles, which have a number of purposes. Storage sacs for cells. They're going to be there for nutrient storage, for physical support. It's also a predator deterrent, waste management, as well as sexual reproduction. Chloroplasts are the plant cell's solar power plant. Okay, they're there to create energy from the sun, and then once it's able to create glucose from the sun, that glucose then has to be turned into ATP in the mitochondria. So we have to consume glucose. Plants are able to make their own glucose. Either way, once you have glucose, we have it from consuming it. They have it from the sun, or plants have it from the sun and from the chloroplasts. Once that takes place, you have to turn that glucose, whether it's from consuming it or if it's from the chloroplast, you have to turn that into ATP, and that happens by mitochondria. And then lastly, this is a nice little diagram they give you in the textbook, and it goes through just the basic functions of the different organelles that we have discussed, and it also tells you if it's found in an animal cell or a plant cell. So you see here, look, cell wall, not found in an animal cell. Chloroplast, not found in an animal cell. And vacuole, maybe, but usually they're very small. They're not going to be big. If you see a really big vacuole, that's going to tell you it's a plant cell. So keep these things in mind as we move forward and we continue to really build on our knowledge of biology.